Welcome everybody to today's webinar, Why DNA Quality Matters from Research to GMP. And thank you for joining. My name is Dr. Marcello Stein and I'm the Commercial Director at PlasmidFactory and will moderate this session. The webinar will be recorded and made available to all registrants afterwards. Participants are muted but can post questions in the chat using the question mark symbol in the upper right. Questions are only visible to the moderator and will be read out at the end for answering. You can also download additional material using the material download function, again in the upper right. Please adjust your viewing window in the center according to best visibility using the zoom buttons. It is now my pleasure to introduce today's speaker, Dr. Martin Schleef. Martin is the founder of PlasmidFactory and served as its CEO for over 20 years, shaping the company into a leading specialist in high-quality plasmid in Minicircle DNA. He studied at the University of Bielefeld, completed a postdoctoral fellowship at the Institut Pasteur, and spent 5 years as a group leader at QIAGEN before founding PlasmidFactory. Today, he continues to support the company as a board member and advisor, contributing his deep expertise in DNA manufacturing and gene therapy applications. So Martin, the stage is yours.
Yeah, thank you, Marcello, for the invitation for today, and welcome to the audience. I structured today's webinar into these parts. First of all, I would like to talk about the role of DNA vectors in cell and gene therapy. I would like to talk about the influence on the quality caused by the sequence and the structure of the plasmid. Will explain how DNA production in high quality works today, and talk about also about the robustness of this and the quality control afterwards, which is important. And then I would like also to introduce you a little bit to our top research product, which is the Minicircle, a new kind of plasmid with certain advantages that I would like to explain you. And this is the idea. And my the first section that I would like to talk about is why plasmid DNA, because you know plasmids from your research. Plasmids are used initially to make viruses. Plasmids can be used as they are. In general, they are DNA and they are used to transfer genetic information into cells. And the job of PlasmidFactory in that field is to make them in a way that they are always in the same quality. Of course, additional tasks are to further development these vector systems, which we do. And the idea behind is that research DNA can be used through all stages of work, which is research, standard research, classical basic research, but also preclinical and then clinical research, maybe up to market supply, because this is then in GMP. And if the quality is the same from beginning to the end, this is something which is extremely helpful and necessary to be able to use all the data that you generated before also in clinical applications. And our experience in this is coming from more than 3,500 large-scale productions over the last 25 years. And the field where it's used is the cell and gene therapy. The cell and gene therapy is an area where certain instruments are used. I called it a toolbox, and the tools are cells. Which cells? It's a patient-derived cells or eukaryotic cells, which are immortalized to do research with, but also other cells, the cells that we are using, those E. coli that are helping us to make the high-quality plasmid. So it's both cells, but the one cell is helping us, the other cell is the target of the product that we are producing, and the tools to get them into these cells are used by you. For example, electroporation or lipid nanoparticles, polyethyleneamine, and all the other instruments, tools, physical applications to get a DNA into the cell and there into the nucleus. And there is another tool listed, viral delivery. If viruses are transformed into viral vectors, then this is something where the evolution had helped these viruses to be so perfect in transferring genes, and this is just used by the system. Also for these, it's necessary to have plasmids to start with to make proper quality viral vectors. And those are, for example, AAV, lenti, and the plasmids themselves, or the minicircles, can be used to transfer the genetic information. And finally, the RNA. RNA is a nice pharmaceutical for transferring genetic information, because after a certain time it's gone. So if you like to remove it after treatment, it's a nice tool. If you want to keep it, DNA is more stable than RNA. So the different formats of DNA are listed here. What you know is the plasmid DNA, the tool for transferring genetic information. But it's possible, for example, to just take out of a plasmid that part that is really interesting for the application, which is a so-called gene of interest, the expression cassette that you want to be expressed in the cell. You can cut it out. And we did this over years, called MITCH. It's a double-stranded DNA, which is covered on both sides by loops to be protected from degradation, linear and minimalistic. And the development that I would like to point out is the minicircle. It's the same. It's also without any backbone. It's super helical because it's quite condensed by the superhelicity of the DNA, like a plasmid, but simply smaller. So the decision which choice of vector in the selection that I've shown you on the left side is influencing the transfection efficacy, the safety, and of course, and this is more interesting for us, the manufacturing feasibility, because we would like to deliver a product where you are happy with. And of course, the regulatory acceptance, if a vector is safe, the acceptance, as you can imagine, is of course a lot higher. How should the vector look like if we talk about plasmids? Typically, plasmids are not identical. It's not a generic process to make them. It's a process that is more or less the same in a way that we have, of course, to start with cultivating cells where we make the plasmids in the cells. We have to take them out. We have to purify them to remove all side products that are also in these E. coli cells. And at the end, it has to be very pure. So this sounds typical. But plasmids are different. They are bigger or smaller. They have certain sequences. These sequences are, let's call them difficult, because they are sometimes even toxic for the cell. So they don't, to say it in other words, E. coli doesn't like them, so they cause problems. The topology is influenced by the sequence. So we have different forms of plasmids, and we have regulatory elements, those that you know, like promoter and poly-A signals and things like this, but there's other elements, and they all influence the form. If you look on the picture here, it shows on the left side of this line, it shows all the parts that are used by us for making the plasmid, but the part that people working in cell and gene therapy, they are using this part. It's a expression cassette. We call it sometimes gene of interest. Also, this is what is needed, and this is needed by us. So it would be interesting to only take the right part, and this is the idea. Also, these needles here are symbols for so-called CPG motifs. And these CPG motifs have a certain disadvantage as well, because they are downregulating the expression in the eukaryotic cells if they are present there. So to have a few of them is an advantage. Coming back to the form of the plasmids, here we have shown in a nice collaboration with the University of Bielefeld and the MDC Center for Molecular Medicine in Berlin, how they look like. We made atomic force microscopy pictures and of course an agarose gel, and we can show here on the left side how they look. This is a supercoiled plasmid in the monomeric form. Below is the dimeric form, and if they are relaxed, they are looking like this. Relaxed means one strand is broken, and this relaxed form is bigger. And bigger means it causes differences in the formulation later if you, for example, formulate transfection aggregates. And this means if you have these different forms and if they are different from production run to production run, this is a clear disadvantage because you have a variation in your product that you are making later with this. Also the size, and this is interesting, the size is, for example, double if it's a dimer, and dimers are happening. So E. coli cells makes predominantly the monomer, but they sometimes also, and this is up to 10%, make the dimer. They shouldn't make the open circle, but they make the dimer, and this is then a larger plasmid. And larger plasmids in general are more difficult to make because the yield is lower. And if the yield is lower, this has a strong influence on the impurity profile. For example, if you have a little bit of DNA, a large DNA plasmid in an E. coli cell and you take it out in a purification process, you have always the same number of bacteria that you are opening to take the plasmids out. And all the other products that are in the cells are there, and you have to get rid of them. So the ratio between the contamination and the plasmid is changing into a direction that is not an advantage for the purification. So we have to take all this into account, and if we couldn't change it, we would increase the costs of doing this. So our task is to improve the processes to be able to get rid of the contamination, and the contamination is RNA, DNA, especially chromosomal DNA, the proteins, the endotoxins, and so on. And then the sequence is a topic. I mentioned it already. For example, those plasmids that are used for AAV production, they contain ITRs, the sequence elements which are flanking those regions which are used to carry the gene of interest if an AAV particle is made. Or for the COVID vaccine production, the homopolymer, a very long stretch of A is there. And this is what E. coli doesn't like, as I said before. And the easiest for an E. coli strain is to get rid of this problem by deleting it. And this is what they are doing. Also, hairpin structures or repetitive elements which are used for an molecular internal recombination shortens the plasmid. Shorter plasmids replicate faster. They overgrow in the culture the right plasmids. And this is what we want to avoid. So in these applications, we try to avoid that this happens. And this is why we are working heavily on improving cultivation technology, purification technology, to allow these sequences, although the cells don't like them, to be made because they are difficult to be produced, and we want to avoid that our clients get the mixture of the correct and the deleted product, because it would look like it's okay, but it doesn't work that good, and it's irreproducible. And this is not a nice situation if you want to use these data, especially for later clinical applications. The mentioned CPG motifs, you remember the needles there, they can be removed. There was an attempt in collaboration with the University of Oxford. We tried to make zero CPG plasmids. okay, it worked, but this is something which makes it more difficult to produce the plasmid. And our idea was then, and I come back to this later with the Minicircle story, to get rid of those parts of the plasmid where these CpG motifs are predominantly present, and they are present in the bacterial sequence part of this plasmid. And one interesting aspect is also, and we see this or saw this for a long time, the typical plasmid that is sent for amplification to us was almost always with ampicillin resistance. This changed, but only a little bit. And our strong advice is to rethink about this early enough, because the change from ampicillin to kanamycin resistance is something which is not difficult. Sometimes even we offer to change this, because the regulatory hurdles with ampicillin resistant sequences are too high. And if you want to go into clinics later, our strong advice is to change it early enough, because then you can use all the data, the starting data of these plasmids for later applications. You cannot that easy change a plasmid if you're on the way to clinics, but if you don't get a permission for clinics because you have the wrong selection marker, this is something which is also a strong disadvantage. And therefore our advice is first to change it, and the second is maybe to completely avoid it. Again, an argument for the Minicircle that I will come to later today. So the negative consequences for the cell and gene therapy could be irreversible results. As I mentioned, if the batch is inconsistent, this would be a problem. Technology transfer difficult means that if you have a technology that works in small scale and you want to have it in large scale or even in pharmaceutical environments, it's really tough to overcome those problems. It's time intensive, it's cost intensive, and it's delaying everything. And the worst case is a late stage surprise, because if you have to change something, if you're about to go into clinics, this is really not nice. And in our production facility, we have experience with these difficult plasmids. We are optimizing for each plasmid the production process and can reproduce it easy at a later stage if additional DNA is required. And we have an analytics which is developed by ourselves in collaboration with our academic partners, being able to analyze the quantity of the different forms of the plasmid that I mentioned, to look for all these features that I summarized in the beginning that could have an influence on this. And this is an absolute requirement if you ever think about going into GMP with such a product. I would then like to show you with some pictures and some explanations how we do the production run. It's important not to see that at the end the QC is nice and everything is running smoothly. You have to go through a process where you, before the start the process, are sure that the product will be as expected. So everything should be reproducible. We start with a pilot run before to discuss about the outcome with our clients. We do this cell banking, where we have a cell bank that is also there if another production run should be needed in the future. And if the cell bank is there and the cultivation starts, it depends a little bit on the scale that is required. Typically, small scales are needed. In between, especially for certain applications, larger scales are required, and very large scales were required for making mRNA for the COVID vaccine. Therefore, we have built a facility where 10-gram batches are possible in this manufacturing. And if the scale of the fermenter is not large enough, our R&D department developed some ways to use a certain volume with a higher yield, which is then fed batch cultivation and other approaches in that direction, to be able on a certain volume to increase the productivity significantly. But then always the first question that we see is, how clean is such a stainless steel fermenter after making one plasmid? What about a second plasmid? Is there a cross-contamination? And we analyzed this here on the left side, where we removed any potential contamination and on a basic level. So it's possible to remove this potential cross-contamination from steel fermenters. But on the other side, it is state of the art, at least in eukaryotic cultivation, to use single-use fermentation. And in a collaboration with Sartorius, we analyzed if it's possible to also have a cultivation in single-use fermenters, which we successfully could do together. And here on the right side, it is shown that stainless steel fermenters and single-use fermenters show the same result if transfer plasmids carrying difficult ITR sequences or very large plasmids, a helper packaging plasmid for AAV production. Also here, no difference in the quality and the quantity between both of them. And if we are working in large scale, it's always important to be able to handle such huge amounts of biomass. And therefore, we developed an automated system where we have an inline lysis, and at the end, if you do this alkaline lysis, which you maybe know from kits, where you shake everything if the buffers are mixed with the bacteria, and at the end there is something floating on it. And here this crystal clear liquid is cleared lysate that then is used to go into the chromatography. And a first part of the bacterial contaminations are here. All the chromosomal DNA, the denatured proteins, it's all there. Here in this liquid, you have the plasmids that you want to have and certain other contaminations that then have to be removed by the chromatography process. And this is something which is not only the QC at the end, also the in-process controls are relevant, and therefore we made some studies here to show how we removed these contaminations from the E. coli cells, the chromosomal DNA, RNA, proteins, and endotoxins. This is done with different chromatography steps, and you can see in this picture that here you have some smear of degraded chromosomal DNA, and sometimes you have also RNA at that position, but typically lower. And this is gone, and you can see here with quantification of the chromosomal DNA that after some time and early in chromatography, you don't have this anymore. You can do the same with protein, and it's important. And therefore, you can see that there is already in an early step of chromatography, this is all gone. So then we are focusing on separating the intact from the non-intact plasmids as well. And if we only have super coiled monomer and maybe a little bit of the dimer, this is something which is quite reproducible to be used in a later application step. So this is in-process control, and the question is, how reliable is the process? This is also important, because if the process is depending on luck or not, this is not really professional work. So we try to make sure that the process is constant, and therefore we did this analysis of some production runs with always the same product, where we could show over months that the OD of fermentation is on almost the same level, and also the total amount of biomass. We did the same also for other parameters like the quality of the DNA, the content of DNA, and so on. So this is all not changing. And this is very important, because this shows that the process is robust. And if we would see any change, then this trending would be used to involve our quality assurance department to find out why is it different. So this is something which is important to be done, to be sure that there is a constant quality. And the final quality can be analyzed then with such instrument. It's a SciEx instrument, and the collaboration is together with the University of Bielefeld again, where this technology together with us has been developed. And it's possible to separate between the supercoil, which is this peak, and for example, the open circle. And there is always a baseline separation. Here, for example, is the dimer peak, and we spike this with different forms of molecules, other plasmids as well. And it's always possible to see this. The linearity and repeatability have been shown as well. This is all necessary to validate quality control assays to be sure that we can claim that the product is identical from one to the other manufacturing run. And not only the process should be stable or robust, also the product. And therefore, the question was, how stable is the plasmid if we store it? This was a question of our customers, and which buffer should we use? And in the old Maniatis literature, you can see TE buffer is what you have to use. But in TE buffer, there's TRIS, there's EDTA. And we thought about what is with water for injection, because this is almost nothing except of water. And we tried it with water for injection at minus 20 degrees Celsius. And you can see that there was not any influence on the stability for the monomer. It's not degraded. And therefore, we think if it's at minus 20 without changing this temperature, very constant, then the DNA is safe and stable. And the other question was, what happens if we thaw it? So what we did was an analysis where we did over 2 months certain circles of freezing and thawing. And we did this 16 times. And the OC band is just at the end. There is a little bit of this coming. So it's more robust than we think. But of course, it should be stored at lower temperatures, at minus 20, minus 70 is not necessary, and not freeze thaw them too much, but a couple of times is something which is, of course, acceptable. So this was all about plasmid DNA, because we wanted to make sure that this product is maintained stable at our customers if it's delivered, and therefore we investigated some of those aspects where we received questions for. So now I would like to talk about the other plasmid, which is the Minicircle, and summarized here those forms that are existing so far. As I said, the plasmid, circular with backbone, of course, this is what I said. But this is the disadvantage, that it has this backbone, and the idea was to make it smaller. This is existing. There is certain constructs called mini or nano, whatever plasmids. They have a reduced backbone, but they have a backbone. It's only smaller, which is good. It's the right direction, but it's not really the goal. The other idea was what I mentioned before, this stuff like doggy bone and our midge. This is a linear form. It does not contain any of the backbone. This is good. And it has caps on both sides protecting it from degradation, but it's not super coiled. And therefore, the idea was to make a plasmid-like molecule, super coiled, circular, completely independent from being degraded by an exonuclease. So this is the summary of what is existing. This linear molecule is here, the midge, and it is something which is produced by using enzymes other than the plasmids that we are using. And therefore, this is nice, but we wanted to further improve that system into something which is circular and supercoiled and therefore very small. And if it's very condensed, like a supercoil it is, this means that it's small enough to get into the cell and into the nucleus. So this is a summary that you can have a look on later if you like, but the important aspects that I would like to point out is the lower DNA toxicity, and therefore that it's so small, we have a higher transfection efficacy. Also, there is no cargo size restriction. So we have made mini circles where I'm not sure if the word minicircle is the right word, because it is 22 kb, which is really not mini, but it's without antibiotic resistance and selection marker and origin of replication. So the backbone is completely gone there, and this is good. And this has been shown, that, for example, by higher efficacy, shown here with luciferase, and also we don't have this backpacking effect that is visible in AAV. So what happens is that you remember the plasmid that I have shown you. This part is only needed by us making the amplification of a plasmid, and this is a gene of interest. And by adding recombination sites here and an intermolecular recombination, it's possible to end up with a minicircle and the mini plasmid, the one that we do not want to have. Now we have to get rid of the mini plasmid. We get rid of the mini plasmid here. This is the process in the cultivation. You see, we start with a parental plasmid, the big one with the recombination sites on both sides, and then we do the recombination, and it starts at minute 1. And after 10 minutes, it's almost done, and after 50 minutes, it is done. So we have the two circles, the mini circle and the mini plasmid there, and now we have to separate these two guys. It's a process that is strictly unidirectional, extremely fast, and it's happening in the cell, in the fermenter. And from there, we go into affinity chromatography, where we at the end have only one product, which is the mini circle, and with the CGE that I mentioned, we can analyze that it's a pure mini circle without any other topology and without the other circle included. And then we thought that maybe we can use this for applications where it's important to use a DNA vector that is not bad for the cell, to say it in easy words. So the reduced toxicity is something which we thought could be helpful, and in a collaboration with partners in Germany and Europe, we had tested this, and here is a publication of the application of a minicircle in combination with Sleeping Beauty transposon system with the lentiviral approach of a CAR T cell therapy, where it's possible to obtain the same results as with lentivirus, but the difference between lentiviral vector and minicircle is that we have here the integration in a random way, while the lentiviruses integrate into the actively transcribed chromatin, which is the genes. And in the analysis of this publication, which is a great publication, you can see that the maximum of distribution, which is defined here with the gray bar, is not reached, but it's close to this, while with lentivirus, the integration into the genes is with a higher likeliness. So the factor of safety is higher with the minicircle, and also the part of the minicircle that is used where the transposon is jumping from into the chromosome is not in the drug product anymore. So this is something where the safety of this type of vector is shown. And the false packaging in AAV production has been shown as well, where in comparison between a two plasmid system and a two minicircle system, the titer of the AAV viruses is significantly higher, which is a cost effect, of course, and if not the wrong sequence elements, which is the bacterial backbone, is packaged into the AAV. This is also good, because a patient doesn't want to be treated with the backbone of the plasmid used for production. And this is what we have published in molecular therapy some time ago, where we could show that if plasmids are used for making AAV, the level of wrong packaged is quite high. If we change one by minicircle, the other is still a plasmid, or the other way around. This is already nice if the transfer plasmid is a Minicircle, but the nicest case is this one, where we don't have any wrong packaged sequence of the backbone, because there is no backbone that could be packaged. Therefore, we don't have this in this batch of viruses that have been analyzed. And the people making AAV see a further advantage. We call it ITR protect and ITR rescue. The ITR sequences are protected from being damaged. This is a collaboration again with the University of Bielefeld, where we found a way to produce these plasmids without affecting the ITR. The most ITRs that we receive here for production are damaged, and in some cases we even repaired them. So this is something which is possible because the yield of making AAV is significantly depending on the intactness of these ITR sequences, and this is something where we put the focus on and where we are happy that it's possible to offer this service in between for our customers. And this is why I would like to summarize the experience that we have here and to show you in this picture our team. And I would like to thank this team to be able to say today that we have a success rate of 99.9% for making a plasmid, because sometimes it starts difficult, but they try everything and they are able to, I would say, to make any plasmid. If it's possible to make it, we can make it, and so far we did. And I'm very proud about this team. And to give you a short overview on the quality levels that we in between established, from scientific quality over the high quality up to something which just was built and is operational, our GMP production facility shown here in this picture. And the first projects are finished. We are happy that everything works and that we are able to supply this. And this is the first GMP production facility for Minicircles. So you remember the Minicircles with the production technology that I just presented is done here. And it's now possible to produce this interesting type of vector in that facility, and it's used already, and clinical trials are ongoing, especially with Minicircles. There's an overview that you can have a look to with Minicircles that are successfully used in these clinical applications. There is more than what I summarized here, and we are happy that just in time we have finished this facility to be able to support such clinical trials for these Minicircles, being able to treat cells that are sensitive and therefore need Minicircles, because it does not work with plasmids and it's not that safe with viral vectors. So I'm at the end of my presentation. I wanted to want to summarize that it's important to think about the quality early enough, to talk about it early enough, to start with research material that is produced in a way where it's also possible later to work with us as a GMP partner, because you don't want to change the partner. You want to start with us and want to go with us, and we would like to go with you to support you with your projects from beginning on. And maybe you like it because we like to comment. And if we have an idea where we think we would love to give you the idea to improve your system, we do this. I hope that you like this information that I summarized today, and would be happy to answer any questions that you may have about this. Important for me is to say that this GMP, which was my dream, is the truth now, and in our case it's a full GMP. We don't talk about something like GMP or a little bit GMP. It is full GMP, and it's possible to make these products there that can be used in clinics and further Yeah, thank you for listening to me. And if you have any further question in the future, you can ask it now, but if you have it in the future, let me know. And I hope to bring you in contact with the right people of the team to answer the question and to start early enough in talking about these projects. Thank you.
I have also a small request at this point. For latest news and regular updates on our products and services, follow us please on LinkedIn. This is always helpful. Of course, like Martin said, feel free to reach out anytime for additional questions and requests. The first one is, how does the Minicircle DNA actually impact viral vector production, for example, in AAV?
Yeah, in a way, it is, as I said already, that this wrong packaging is avoided and the yield is higher. So we have two advantages. If we combine them, and if we then also combine this with the intactness of the ITR, which we can make if it's not there. So we have three advantages together. And if we combine everything, and we did this in our R&D group already, then we can see that we have a great vector that is able to significantly increase the productivity with having no wrong sequences packaged deriving from the bacterial backbone in high yields. And therefore, I think that this is the most important advantage of the Minicircle in AAV production. It can also be used for other virus production strategies, but this is the main advantage that we identified together with our collaborators.
Yes, thank you, Martin. A second question just came in. If the Minicircle is actually already clinically relevant, or is it still mainly a research tool?
Yeah, they have been used. We have, for example, a very good collaboration since a long time with the University of Würzburg, with Michael Hudecek. They are using our Minicircle. They have used it for clinical trials. There is other clinical trials ongoing. I've shown you on one of the last slides a summary of applications where it's used, and there is further clinical applications on their way, also for other diseases, not only a CAR-T application, also other cells like stem cells, very sensitive. This is something where the Minicircle is able to overcome the problem that the sensitivity of these cells causes the cell death. And this is where I'm sure that further studies will come, and we are prepared with the facility, and we started already some projects that will end up in the facility.
Yes, thank you. Yeah, similar question also here. In gene-modified cell therapy workflows, where does DNA quality typically become a bottleneck?
Yeah, the sensitivity is one aspect that I mentioned already. If the cell is sensitive, it's suffering from the DNA, but mostly it's also a byproduct could be potentially a risk. The plasmid itself carries sequences, as I said, that the cells don't like, but there's also a chance of having still contaminations inside. This is where we are always fighting for. I've shown you that the chromosomal DNA is removed. This is very important because chromosomal DNA is extremely big. And in all cases where people use transfection agents, where they want to make complexes, a chromosomal DNA, which is an extremely large molecule in comparison to the plasmid, is influencing the complex building. And this causes wrong sizes of the complexes or inhomogeneous preparations that are then used. And this causes irreproducible results in all the preclinical studies. So this is something which should also be taken into account. And if chromosomal DNA is removed, then it's not a little bit or a little bit more or a lot chromosomal DNA in the plasmid. It's just not there. And not there is what our goal is, to make sure that only plasmids are participating in building a complex, or minicircles in participating a complex, that is then used for transfection, or if lipid nanoparticles are built, this is also something where you need a homogeneous DNA as one of the components that is added to the other elements of such a formulation. Otherwise, you end up in irreproducible products being made with DNA, and this cannot be successful.
Thank you, Martin. There's still another question waiting. So how much does DNA purity and topology really influence biological performance?
Yes, this is something which we thought about, and we had a nice collaboration with the University of Munich, where the question was, does a monomer have an advantage in comparison to a dimer of a plasmid? Because dimers, as I said, are typically present in E. coli cells. And what we tried was to make a pure preparation of a monomer, which was possible, and a pure preparation of a dimer, not that easy, but we made it. And then we compared the monomer with the dimer in their performance. And it could be shown that, as expected, the dimer, because it's bigger, was not that easily be transferred into the cell, because we have two checkpoints where we have to get into the cell. First is the cell itself, and the second is the nucleus, where the DNA should be in the cell. And this was more difficult for the double-sized plasmid. But as a surprise to us, it turned out that the double-sized, the dimer of the plasmid, was more successful in expressing the genes. We didn't further investigate in this, but it was so significant that we have the idea, this is a hypothesis, we didn't further check, that the transcription of the genetic information on this is by the transcription mechanism easily made if everything is on one molecule. So let's say it's closer together. I don't know if this is the real reason for this, but we are happy that a dimer is in a way equally active than a monomer, although it's only up to 10% in a preparation. But we could clearly show that if this molecule is an open circle one, that then the success is significantly reduced, and it's statistically damaged, because open circle is caused by mechanical shear forces killing one of the two strands, and this means it's not condensed anymore, it's relaxed, and even worse, if the nick, which is the reason for one strand breaking, could be in the gene of interest, this is even worse, because then this gene is not fully transcribed, and this means it doesn't work, and efficacy is significantly reduced by this. So the topology has a strong influence on this, and this is true for all types of transfection. It's the chemical transfections that I mentioned by building complexes, but also electro-gene transfer is something where this is affected, and all other approaches maybe as well, because the negative influence is happening on the level of entering the nucleus. And therefore, I think if everything is CCC, this is the supercoiled form, then you can compare results, and you cannot compare them anymore if you have a mixture of the intact and the damaged plasmid in your preparation.
Thank you, Martin, meaning that the impact is quite substantial, actually. And so best quality DNA starting material is more than advisable.
Yes.
Good. Yeah, thank you. For now, I don't see any additional questions in the chat here. With that, thank you again for this interesting talk, Martin. We can close the session here. And as said before, every registrant will receive the slides and the recording of this session. And as always, feel free to reach out, contact us in case of further question. So thank you for listening. Thank you for participating, and have a great day.
Thank you. Bye-bye to all of you.